So in this next screencast, we are going to look at errors in meiosis, things that can go wrong that affect these chromosomes. So the first, to introduce this, I want you to take a look at this karyotype and see if you notice anything interesting or different or unique. Anything? Anything? Take a look at chromosome 21. Whoops. Chromosome 21, it turns out has 3 chromosomes. And so, yeah, how does that happen, and then what effect does it have? Well, let's take a look. So the first type of error that can happen in meiosis is a change in chromosome number. And the cause of this is something called nondisjunction. And nondisjunction simply means that there's been an error in the separation of chromosomes during meiosis. And note, this could happen during meiosis 1 or meiosis 2. In the case of meiosis 1, basically the both pairs of homologous chromosomes, instead of getting separated out, they've just, for whatever reason, accidentally both get pulled to one side. And so the result is that this product has 3 homologous chromosomes, and this one only has, or 3 pairs, and this only has 1 pair. And so you have these gametes with an extra copy of a chromosome or missing a copy. It can also happen in meiosis 2, where the sister chromatids don't go to the right, don't separate properly. And in that case, again, you get n+1 and an n-1 situation, and then these are going to be normal gametes. So if things don't separate properly, that can result in this change in chromosome number. And so examples of this, aneuploidy is where you simply have an abnormal chromosome number. So instead of 46 chromosomes, you have a different number. And so one example of aneuploidy is trisomy, where you have this extra copy of a chromosome. So the example that we opened with here, where you have an extra copy of chromosome 21, is called trisomy 21. You may have heard of that as Down's syndrome. Down was actually the doctor who first described this condition, and we're sort of shifting away from that language because it's, I don't know, there's just something about, Down owning the syndrome or something that's weird. So we sort of call it trisomy 21. And then over here on this diagram, you can see all of the consequences of trisomy 21. You certainly don't need to memorize all of these, but you can see that they're pretty significant. There's significant mental retardation. Brain development does not happen typically. There's some just physical features of broad, flat face, slanting eyes, short nose. There's some heart issues associated with it. There's just lots and lots of issues. And basically the reason for this is because if you're getting an entire extra dose, so to speak, of all the genes on that entire chromosome, that is going to disrupt homeostasis in the body. You're just having too many protein products and a lot of things, and that causes a lot of problems. Another example, Klinefelter Syndrome. Here, it's the sex chromosomes that don't separate properly, and so you end up as an XXY individual. Now, because of that Y chromosome, you do have the genes that trigger the development of male behavior, but because you have that extra X, you also get some feminization that happens. And so there's even though you're male, there's some breast development that happens. You have sort of the female pattern of fat storage in the hips, and tendency to grow less hair on your chest, poor beard growth, etc. So it's more of a feminized male that results from this. Another example is monozomy, where you're missing a copy of a chromosome. An example of this is Turner's syndrome, and in this case, you're missing a copy, one of the sex chromosomes. So somebody who is Turner's syndrome is always going to be female, and that's because the X chromosome, remember, has about 2,000 genes versus the Y chromosome, which has about 100. So if you're missing that X, you're missing too many genes to survive. And so the embryo will never be able to develop fully. So the only way you can survive this way is if the one chromosome you have happens to be an X, in which case you don't have the Y, in which case you develop female. So anybody with Turner syndrome is going to be a female. Again, you don't need to memorize all the symptoms listed here, but in general, one of the major ones is that the ovaries don't fully develop, and so then there's some sexual development issues. Menstruation doesn't happen, there's poor breast development. There's some other things, the aorta, major blood vessel leaving the heart is constricted, and things like that. Note that, these three examples we've talked about, examples of this are actually fairly rare and usually involve smaller chromosomes. So there's trisomy 21, there's trisomy 18, there's a number of conditions related to the sex chromosomes. You never see trisomy 1 or monozomy 2. Think about why that might be. And the answer is, it's because there's just too many genes on these larger chromosomes. So if you have an extra copy, or you're missing a copy of one of these larger chromosomes, then the changes are so disruptive that the embryo simply cannot develop and dies, likely even before the mother would know she was pregnant. So even the ones we do see where they're the smaller chromosomes, 21, 18, those are, pretty significant issues arise from those. And so basically an extra copy or missing a copy of a chromosome is a pretty big deal. Another way that you can have a change in chromosome number is polyploidy. So here, instead of just an abnormal number of chromosomes, you have more than 2 complete sets of chromosomes, right? So haploid means 1 copy of each chromosome. Diploid means 2 copies of each chromosome. Polyploid means many copies of each set of chromosomes. And so you can see, they've got, I don't even know, is it 3 sets? 6? 6. There's 6, right? 2, 4, 6 sets of each chromosome. So this is actually common in plants, but rare in animals. And the reason why is because plants are much more flexible in their development. A human, for example, we've got 2 arms, we've got 2 legs. The heart goes here. Everything is kind of fixed in place to a very specific plan. Whereas plants, a tree can just kind of grow and I'll sprout a branch in this direction, and oh, there's some light, I'll bend this way. And so they're just more flexible in their development, making these extra copies less harmful to them. Polyploidy does occur naturally, and lots of agricultural products are polyploid. So humans have sort of found these polyploid varieties and said, hey, this is a good quality, let's use it. So, wheat is actually 6N, tobacco 4N, don't smoke. Potato 4N, bananas are triploid 3N, boysenberries 7N, strawberries are 8N. So there's 8 copies of each gene. We actually, in Gender Enriched Bio, maybe you did a lab where you isolated DNA from strawberries. We choose strawberries on purpose. It's 8N. There's a lot of frickin' DNA in that little berry, and so it's a great specimen if you're trying to isolate DNA. So why are polyploids useful? Well, they often have larger fruit because the cells themselves are actually larger. And so, here's like a typical strawberry that's 2N, and then here's a polyploid strawberry that's 8N. And you can see these pictures are quite dramatically different. Grapes, 4N are larger than 2N. So they're useful for that way. People like larger fruits. One example, kind of a tragic example of a polyploid is the Cavendish banana. So the banana that you typically eat is triploid. And I think it's worth watching this little video clip. The Cavendish banana is in trouble. It turns out that these bananas don't have seeds. And that's because they have an odd number of copies of chromosomes. You can imagine if at metaphase 1 of meiosis, if you line up 3 copies of the chromosomes, you can't split evenly, right? 2 are going to go one way, 1 is going to go the other, and so then the gametes aren't going to be able to recreate the 3N necessarily. And so they're sterile. And as a result, they have to be reproduced through asexual means, and that means that pretty much all Cavendish bananas are genetically identical. The risk of that, of course, is that if a parasite is able to sort of figure out how to attack and destroy that banana, it now has this whole field of genetically identical individuals that it can take out. And that's kind of what's happening with our banana. It happened before with a different variety of banana, and now it's happening again. So that's a very interesting link there. Boy, Hold tight to your bananas, ladies and gentlemen. Another type of error that can happen in meiosis, so we just went through changes in chromosome number. Another type is changes in chromosome structure. So the cause here is an error during crossing over. So crossing over, remember, happens in prophase 1 of meiosis, and so just little bits of DNA are swapped. Well, there's different ways that mistakes during crossing over can result in a problem. So in a deletion, what happens is you're missing a little segment. So this little segment, it's supposed to cross over to another chromosome, but it gets lost. And so instead of 1, 2, 3, 4, 5, it's 1, 2, 4, 5. You've lost a little piece. Duplication is where you cross over, but instead of transferring this to another chromosome, it stays, but then picks up another piece. And so you have like a doubling of that. An inversion is where a region breaks off, and then it reattaches in that reverse order. So instead of 1, 2, 3, 4, 5, we have 1, 4, 3, 2, 5. And then a translocation is where part of a chromosome breaks off and then reattaches to the wrong other chromosome. So all of a sudden we have 1, 2, 9, 10, 11, 12. So to understand these, I think it's helpful to take a look at our models. So let's pause for a moment and do that. So imagine we're at prophase 1 of meiosis, and we are going to exhibit crossing over. Well, normally these would just swap, and that'd be the end of it, right? But if there's an error during crossing over, then something bad can happen. So a deletion would be where we're going to cross over, and we transfer this one, but then this little piece, whoops, for whatever reason, it doesn't attach properly and is lost. We now have a deletion. A duplication would be where we're supposed to cross over, but instead, oh, that one doesn't leave. And then this one kind of sticks in there, and now you've got this duplication and a deletion over here. An inversion would be where we swap over here, but this one gets inverted. And so now the order of the genes is off, which again can cause problems. And then a translocation would be where we break off, this one attaches, but then this one somehow gets lost. But then, oh, what's this? Our Milky Ways. It just kind of gets attached to that chromosome kind of randomly. And so that's called a translocation. Examples of changes in chromosome structure, there's a condition called PreDucha, which is a deletion. And again, it's a very specific deletion in a very specific location. It happens in chromosome number 5. And we just lose this little tip of the chromosome. So this chromosome is a little bit shorter. And it's called PreDucha, which is cry of the cat in French, because the voice box where your vocal cords are housed is misshapen. And so when the babies cry, it actually sounds like a cat, I guess. I've never heard the sound. There's other issues. The head is smaller and the brain development is inhibited. Another example is what's called a Philadelphia chromosome. These actually arise during an error in mitosis, actually, of dividing white blood cells. And this particular translocation, there's this little mistake where this gene kind of gets translocated over here for whatever reason. And when these two genes are put together, they actually make this novel protein that signals cells to continually divide. And if you are continually dividing, what is that going to cause? That's right, cancer, specifically leukemia in the white blood cells. So one form of leukemia is caused by this genetic mutation, this mistake during mitosis, actually, of the dividing white blood cells that creates this kind of unique novel chromosome. So we will pause there and finish with one more screencast.